Hello. Today I will be talking about the endophillin protein family, a family of membrane curvature sensors and generators. As a starting point for this video, I would like to begin by showing a picture of a budding vesicle in endocytosis. The figure shows the cytosolic side of the membrane. As can be seen, a budding vesicle shows various regions of different curvatures, including positive and negative curvature. For example, the neck region of this vesicle is a thin tubule with very high curvature. Such spatial variations in membrane curvature is not thermodynamically spontaneous, and yet this phenomenon is physiologically ubiquitous in all organisms. The spatial variation in curvature is stabilized by curvature-generating and sensing proteins. In fact, one special class of endocytosis, called clathrin-mediated endocytosis, is associated with a variety of biomedical pathways, such as the regulation of LDL cholesterol and synaptic vesicle recycling in the nervous system. Perhaps the most fascinating curvature sensor and generator is endophillin. Endophillin has an SH3 domain and an N-bar domain. Shown here in the ribbon representation is the N-bar domain, the domain responsible for curvature sensing and generation. Primarily a bundle of alpha helices, endophillin N-bar displays the characteristic banana shape of curvature sensing proteins and is dimeric biological assembly form. However, endophillin has one of the highest intrinsic curvatures of curvature sensing proteins. Because of its high intrinsic curvature, endophillin plays a role in stabilizing regions of very high curvature, such as the neck region of a budding vesicle mentioned earlier. At its arch region, endophillin contains a patch of positively charged residues. This is a common feature of curvature sensing and generating proteins. This positive patch of residues enables the protein to have favorable electrostatic interactions with the inner leaflet of the plasma membrane, which is typically negatively charged. These interactions not only favor binding of the protein to the membrane, they are also the driving force for the imprintment of the protein's curvature onto the membrane. This process is known as scaffolding. Scaffolding is one means of curvature generation. In addition to scaffolding, endophillin is able to generate membrane curvature through amphipathic helix insertion into the membrane. Amphipathic helices generally don't insert evenly into the two leaflets of the bilayer. When they insert, the surface of the inner leaflet is expanded relative to the outer leaflet. To accommodate the difference, the membrane bends. The endophillin dimer has 4 helices that can generate curvature in this manner. The helix on the distal arm is called the H0 helix. The helix located beneath the arch of the protein is called the H1 helix. Simulation studies investigating the insertion of the H1 helix reveals interesting properties of these insert helices. The following plot shows the insertion depth of each residue. The yellow region shows the approximate region occupied by the bilayer. Polar residues of the helix are found at the edges of the bilayer, where they can have favorable interactions with the polar head groups of the lipids. Moreover, most of the hydrophobic residues, denoted in gray, are found in the lipid tail part of the bilayer, exactly what is expected when one considers the entropic hydrophobic effect. The H0 helix, however, is arguably the most biologically significant one. Firstly, measurement of fluorescence emission anisotropy experiments with isolated H0 helix peptides show that the helix preferentially inserts in a negatively charged membrane. Emission anisotropy is a measure of how different is emission intensity at different polarizations of light. High anisotropy is characterized by lack of rotational freedom of a molecule in solution, precisely what one would expect if a linear peptide inserted into a membrane. Also, fluorescence was also used to quantify binding by the endophillin N-bar domain and H0 peptides to vesicles of different radii. Intuitively, the smaller the radius of a vesicle, the higher the curvature. Both showed almost identical binding and curvature preference. The H0 helix is able to sense curvature and partitions into higher curvature. Finally, tubulation experiments show that the H0 helix is the main driver of curvature generation. Endophillin could not produce tubules of high curvature with a non-conservatively mutated H0 helix. Finally, endophillin is able to oligomerize into supramolecular structures that generate and stabilize very high levels of curvature. Once endophillin binds to the membrane at intermediate surface coverage, it forms a lattice of linear aggregates. At higher coverage, endophillin can generate and stabilize thin tubules of very high curvature. The top picture shows a simulation-derived structure of the supramolecular endophillin assembly. The bottom two pictures shows the experimentally observed assembly. In the blue regions in the top picture are the H0 helixes. The same simulation study determines that the H0 helix is crucial for oligomerization. The following supramolecular structure is crucial for endophilin's biomedical role in clathrin-mediated endocytosis. Endophillin generates the neck region of a budding endocytotic vesicle. Then, through its SH3 domain, it recruits a GTPase protein called dynamin. Dynamin then completes the internalization of the vesicle cargo by scission of the neck. However, it was only able to do so thanks to the generation of the neck by endophillin and its recruitment via endophilin's SH3 domain. Endophillin is a truly remarkable protein, and much work remains to be done to fully characterize its biological role. A potential role for endophillin in other stages of catheter-mediated endocytosis is still a debated issue, and the H1 insert helices are not yet characterized to the extent of the H0 helix. Moreover, a mechanism to bridge the initial linear lattice arrangement to the final oligomerized superstructure has yet to be determined. A lot yet remains to be understood about the endophillin family of proteins, and such knowledge is crucial to fully understanding endocytotic pathways. Thank you for listening.